first, I would like to start by thanking the organizer for this amazing program and my fantastic mentor, Elise. My name is Kyuson. I'm a founder of Empiri. And Empiri is a resident of J-Labs in Houston, Texas. And our mission is to revolutionize the way we treat cancer patients by providing empirical drug response data prior to treatment selection in the clinic. We are a platform company, and currently we are focused on precision oncology, and we're developing 2 products that I'd like to tell you about. But before I go into that, I'd like to tell you a short origin story, as many other founders have done today. About a month after we celebrated my father's 60th birthday, he found some bumps in his mouth that he thought was an infection. Turned out, unfortunately, he had a late stage metastatic cancer. And the doctors told us that there was nothing they could do for him. My father passed away 3 months after without ever receiving any treatment. And 30 years later, we still say the same thing to many metastatic cancer patients. And that's unacceptable, given that there are over 168 FDA-approved anti-cancer agents. And the problem is that there's not a good way for oncologists to determine which drugs will work best for each patient. And that's the problem that we're trying to change at Empiri. As a solution, we developed a novel 3D culture method called eSlices. eSlice assay is a personalized cancer diagnostic test that empirically measures and predicts each cancer patient's response to drugs before a doctor needs to make a treatment plan for that patient. So the way it works is a small piece of patient's tumor, either biopsy or surgery materials collected, and we can run eSlice assay at Empiri and very soon, hopefully, at site of care by using our instrument that will perform the eSlice assay. And then the doctors have the report before they meet with the patient next time. To look at it a little more closely, eSlice is generated by making very thin precision cut slices of live patient tumor tissues. And these are cultured in defined medium and exposed to chemotherapy, targeted therapy, and immunotherapies or any other treatment anti-cancer agents ex vivo. And in 4 to 8 days, our assay can report drug sensitivity or treatment sensitivity of each patient's tumor to monotherapies as well as specific combination therapies. Now, this report can be used by the oncologist to determine the optimal treatment for each cancer patient. Importantly, eSlice has been clinically validated. In collaboration with Dr. Scott Kopatz and Van Morris, who are leading GI oncologists at MD Anderson Cancer Center, we performed 3 validation studies. On the left, you can see is a correlation graph that shows patient response on the y-axis and eSlice prediction on the x-axis. And this diagonal line going across the table shows you that eSlice prediction is highly accurate in predicting individual cancer patient response to particular treatment response. On the right is a table summarizing similar kind of information. What I would like to point out is that eSlice is not only able to predict responders and non-responders, but the degree to which each patient will respond to different kinds of drugs. Based on these strong clinical validation data and the enthusiasm of clinicians who've been working with us, we decided to go to market with focusing on colorectal cancer. Colorectal cancer is the 3rd most common cancer type in the world and in the US. And currently, 53,000 US citizens, or over 631,000 people worldwide, develop metastatic disease and die. Working with Dr. Kopatz, we have identified a specific pain point in care of these metastatic colorectal cancer patients. And our first assay will be specifically tailored to meet this desperate clinical need. The colorectal cancer diagnostic market is rapidly growing. It is expected to reach $2.8 billion by year 2026. And currently, colorectal cancer is the 3rd most common cancer. But according to this recent review in Nature Review Clinical Oncology, it is anticipated to become the most common cancer in the world by 2045. So that means, unfortunately, the need for accurate cancer diagnostic assay like E-slices will continue to grow in the foreseeable future. And here we will generate revenue through 3 different streams. In the first, we will provide E-slice as a clinical assay. And currently, we are working with regulatory consultants to finalize our regulatory strategy, and most likely will involve applying for breakthrough designation of approval by FDA as a clinical oncology test. In the preclinical setting, we are also seeking partnership with pharmaceutical companies and biotech companies to incorporate E-slices into their drug discovery and development pipeline. As you can imagine, a physiologically relevant assay like this can greatly enhance and accelerate their drug discovery efforts. In the same second stream, we are building an instrument called CAL that will automate the E-slice method. This will be the first point of care, self-standing precision oncology, turnkey solution for precision oncology. And we will generate revenues by instrument sales, as well as continuous purchase of consumable test kits for each patient that's tailored for different cancer types. And finally, we're in a position to collect a large set of highly valuable data from patient tumor drug response, and combined with our single cell analysis expertise, we'll have a unique data set that will be of great value to pharma and biotech companies. We are starting our business with colorectal cancer, but E-slice has been tested in breast cancer, lung cancer, pancreatic cancer, brain cancer, and other cancer types, over 400 experiments. And we have clinical champions in each one of these areas. E-slice has been shown to detect interpatient differences or intersample differences to not only chemo and targeted therapies, but also to immunotherapy. In other words, we are positioned to rapidly expand our product lines to include assays in all solid tumor types and capture a significant portion of this growing global cancer profiling market that's expected to reach $162 billion by 2027. As you know, there are many other precision oncology technologies out there, and I would like to highlight the differentiator of our technology to those. To my knowledge, our technology is the only technology that allows intact tumor microenvironment to be cultured ex vivo without disruption and without extra manipulation. And this is important because human cancer is not only composed of cancer cells, but also stromal cells like immune cells and blood vessels and other stromal cells. And in some tumors, those cells can be more than 50% of the tumor mass. And they play critical roles in modulating tumor growth as well as therapy response. So that positions CAL and E-slice in a highly favorable position of having high fidelity to patient tumor, and at the same time, providing the results in a relatively low cost and time. These are some of our competitors in precision oncology space, including those that use DNA sequencing or organoids or other methods. But I would like to just highlight the fact that E-slice has been clinically validated, is compatible with biopsies as well as surgical material, can provide results in a clinically actionable time frame of 4 to 8 days, and it retains the native tumor microenvironment unique to each cancer patient. And soon we'll be able to provide this as a turnkey solution in our automated instrument. Currently, our E-slice assay is at the end of clinical validation stage, and we will be seeking FDA breakthrough designation and clinical trial for the assay as a oncology cancer diagnostic test. And for the instrument, we needed to invent several pieces that were required to fully automate the process. We have functional breadboard prototypes that we would like to refine and integrate with existing technologies to build a fully integrated system in the next 2 years. The patent for our method has been filed and is in the national phase, And we will be filing 3 to 4 new engineering provisional patents in the next 6 months. So far, we were funding through revenue generation and pre-seed money, and we are seeking either seed funding or Series A funding to take both our products to the market as quickly as possible. And Pierre's leadership team brings all the necessary expertise to execute our plan. I'm a cancer biologist with over 25 years of experience and a faculty at Houston Methodist and Weill Cornell Medical College. Dave is a mechanical engineer and in business development management for over 20 years. And importantly, he's a successful entrepreneur who started MLabs and sold it to Test America. Linus is our chief medical officer. He was a faculty member at MD Anderson Cancer Center in GI oncology and expert in clinical oncology. With that, I thank you for your attention and invite you to join us in changing the way we treat cancer patients. Thank you.
Sure. Thank you, Dr. Yoon. Jeff, I see your hand raised. Do you want to start us off again?
Sure. I'm just too quick with the button. I should go on Jeopardy. So thank you for the presentation. I'm thinking about this in the one form that you talked about it as a way to pick an antibiotic for a particular pathogen. You want to pick a chemotherapeutic agent for a particular cancer. I was actually extremely happy that you spoke to the potential for it in drug development as well. The other one that you didn't talk to, is it possible you could apply your techniques using tissue samples from major organs and also predict toxicity of those organs from the different treatments? I'm thinking now in pharmaceutical biodevelopment as opposed to, you know, approved drugs already. So is that one of the things on your roadmap? And if not, why would you not add it?
Yep, thank you for that question. And actually it is. I started with saying we're a platform company currently focusing on oncology. But yes, certainly we're thinking already about toxicology, neurology, as well as infectious diseases, because we can culture, what I didn't tell you, one of the other major differentiator is that we can take these patient tumors and culture them for like 3 to 5 weeks ex vivo. And we were able to do that also with liver, human liver and brain. And so, yes, that is definitely on our roadmap. And that's where we'll go next once we have all this oncology space covered.
Thank you. Mitzi, I see that your hand is raised. Would you like to go next?
Sure, thank you. And thank you for your presentation and my condolences to you about your father. Thank you for sharing your story. So you note that you have a revenue that you're taking in revenue now. So one of my questions is, you know, where is that coming from? And given that you're a potential strategic partner for the pharma industry, have you already been in talks in terms of looking forward to exit strategies?
Exit strategies, not yet. But in terms of revenue and talking, yes. So we actually had a contract for a big project with Astellas, evaluating their new anti-cancer agent, but that kind of fell through during the COVID time. So our current revenue, actually we're providing 2 services, both tumor slices, the e-slices, as well as a single cell sequencing. And so the single cell sequencing part is going better. But we will be making active efforts to contact pharmaceutical companies for e-slice services as well. And during the COVID period, we were really focused on engineering because that's something we could do in our isolation, and we made amazing progress in that regard.
Great, thank you.
Thank you. Gladys.
One question, as you were focused on the engineering during COVID, completely understand that, of course. What is the strategy for the analytics, you know, to your point, as you breaking down the tumor microenvironment, as well as the stromal area around it and breaking down the cells? As you scale, how will you address the analytics around the platform, especially as you start working with pharma and different clinical groups?
Yeah, thank you for that question. And just to clarify that our technology is the only one that does not dissociate or break the tumor microenvironment. So we kept it all intact. And a key feature of our technology is that we use defined medium. So there's no serum, which makes the analytics very straightforward. For example, as an example, I mentioned that we can measure response to immunotherapies, right? We can not only measure changes in the viability of the tumor tissue, but we can also detect interferon gamma secretion. We use checkpoint inhibitors in the tissue. So the tissue can be used for any post-treatment or longitudinal treatment analysis. We can do RNA sequencing. We've done actually single-cell sequencing from the slices at different time points. We can measure secreted molecules, including cytokines and chemokines, as well as performing proteomics. So we're positioned to really help pharmas understand their mechanism of action for novel drugs, in addition to just performing a physiologically relevant screen to optimize their targets, prioritize their targets.
Now that's actually very helpful, and actually kind of leads me to the comparison of the data that's out there, you know, like the Human Atlas and some of the oncology and data that's coming, especially since, like you said, one of the unique features is that you maintain the tissue intact, yep, compared to others that are actually breaking it up and getting down, right, to the cellular level. So
right, okay, absolutely, yeah, and I think that's critical.
Okay, thank you.
Thank you.
Thank you, Dr. Yoon. Dr. Yoon has rounded out our last pitch today, and they were all fantastic. So thank you very, very much. Thank you for all of you who have stayed through this whole time. We have the People's Choice Award now open, and when you go back to the main room, you will be able to find the URL that you can link and decide who you enjoyed most of the 6 that you heard today. And this will be followed by a panel that will start at the top of the hour entitled The Entrepreneur's Journey, Insights and Advice from Women Academic Founders. So please stay with us. It's going to be very exciting, and join us back in the main room so that you can vote. You may shut your video off, and we'll sort of tell you when it's the top of the hour and the panel will start. Thank you so much.